Hello friends, today we'll be going through the topics involving the neuroglia and the meninges. The neuroglia are non-neuronal and highly branched, non-excitable small cells, and by non-excitable that means it doesn't respond to electrical impulses the way neurons do. They are still signaled and in a way respond to electrical stimuli, but they don't conduct any uh, electrical stimuli or service. It makes up 2/3 of the total volume of the central nervous system, and they have a variety of roles such as providing structural, metabolic support, and electrical insulation. And by that, I don't mean uh, electrical insulation like axons, where the myelin will insulate the signal and allow the electrical impulse to propagate faster. That's of course one of the roles, but not these particular cell roles. They insulate the electrical signal of one neuron so it doesn't accidentally involve another one, so it doesn't accidentally fire. So in the central nervous, there's the astrocytes, the oligodendrocytes, and the ependymal cells, and all of those are derived from the neuroectodermal cells. And of course, those come from the ectodermal layer. And microglia are the circulating blood monocytes that come from the mesenchyme, and that's like the only cell that doesn't come from the ectoderm. And the peripheral nervous system have the satellite and the Schwann cells, and those are also derived from the neurocrest cells, so those come from the ectodermal layer. So these are the different types of cells. The microglia are the smallest glial cells, and they have not very many and not a lot of cytoplasm, and they have fine, short, and irregular processes. They're distributed throughout the gray and white matter, and they have small, elongated, and dense, irregular nuclei. As you can see here, they are darkly stained, and normally they are inactive and this says inactive in normal conditions, but it does work when the nervous system is not under attack, and that only occurs when you are asleep. So the microglia will come to clean out the metabolic waste products in the cells of the brain and nervous system, but it only happens when you're asleep. So sleep is pretty important. That's why people who lack sleep hallucinate and they have disorganized thought. It's because their neurons are literally not able to fire and work correctly. So the microglia are specialized macrophages, and they proliferate in response to tissue damage. And they are not interconnected, so they're not bound to each other. Instead, they migrate through the neuropil. Astrocytes are the most numerous and the largest glial cells in the gray matter, and they have many cytoplasmic processes, and those form the end feet. To me, the end feet always kind of look like, those jelly hands back in the early 2000s. I think they were really popular. So the jelly hands would, attach and bind the astrocyte to other cells, and the end feet will cover the non-synaptic regions of the cell bodies, such as the dendrites and parts of the axon, in order to electrically insulate and prevent the electrical activity of one neuron to affect another. And they also form the perivascular feet, which cover the basement membrane of the capillaries and contribute to the blood-brain barrier. The astrocytes will also expand to form the glia-limiting membrane that lines the meninges and the underlying ependymal layer of the ventricles. And the nucleuses are generally oval or irregular, so they don't look like that. Astrocytic processes are reinforced by the intermediate filaments that are made out of the glial fibrillary acid protein, or GFAP, and they can only be stained with a special stain, so a silver or gold chloride stain. The fibrous astrocytes is one of the derivatives of astrocytes, and those have few straight, long, thin processes that are really rich in GFAP, and those are found in the white matter. And then the protoplasmic astrocytes have numerous but short and thick processes with few bundles of GFAP, and those are generally found in the gray matter. Astrocytes are connected to each other by the gap junctions, and they regulate the extracellular ionic concentrations around the neuron by taking up the excess potassium ions that are left behind because of repetitive firing of the neuron, and by reuptaking all of those like waste products, extra neurotransmitter, it will limit the spread of the neurotransmitter, so that can't spread and affect another neuron or another synapse. And it also mediates the exchange of metabolites, so it will take in nutrients and dispose of waste. And they also work to guide and provide structural support to migrating neuroblasts. And those will, also, migrate in times of growth or in times of damage. And these astrocytes proliferate to form an astrocytic scar during gliosis after damage. And it is theorized that they play a role in intellectual difficulties. And it could be because the end feet don't adhere well, and the mediation of electrical signals isn't occurring correctly, and the reuptake of neurotransmitter might be faulty. So here is the normal physiology. This is the abnormal. So the blood-brain barrier is made of capillary endothelial cells, the basement membranes of those cells, pericytes, and perivascular astrocytic feet. The capillary endothelium is the main component of the blood-brain barrier. It's non-fenestrated, which means it doesn't have any openings or little windows, and they are bound together by tight junctions. The basement membrane surrounds the endothelial cells and the pericytes, and the perivascular feet will cover the basement membrane. So as you can see here, these are the little feet. Here's the pericyte, here's the endothelial cell. This is the vessel that's encirculated, your neuron, axon, and then microglia. Here it is, with the feet attaching to everything. And this is the blood-brain barrier. We'll talk about what goes through it, but once again, here's the basement membrane and the pericyte, and above is deep to it is going to be the astrocyte, the microglia, and the neuron. The blood-brain barrier provides the central nervous system with constant biochemical and metabolic environment that is important for just normal functioning. Some infective microorganisms can pass through the blood-brain barrier by releasing cytotoxins that have a toxic effect on the tight junctions. So with these junctions not adhering the cells together, they can slip through. Such microorganisms can be the spirochete bacterium, or in particular the Borrelia, and that causes Lyme disease. Group B, Streptococcus, that will cause the bacterial meningitis. Treponema pallidum, it's a this is not a bacteria. This is a it's not a parasite. It's a sorry, give me a second here. It's a spirochete, and other substances can pass alcohol, the tricyclic antidepressants, chloramphenicol, sulfas, tetracyclines, those drugs. Those are the most common drugs to treat bacterial meningitis for that reason. And indirect bilirubin provided mostly by neonates and newborns. That's why, remember we were learning about how all those deficiencies can cause a buildup of the uh, unconjugated bilirubin, and that's why it can cause mental delays, developmental delays, is because it can go through and enter the blood-brain barrier and affect the brain. The blood-brain barrier is not found in the choroid plexus. The circumventricular organs are near to the hypothalamus, and those have neurosecretory functions, and those will monitor the concentrations of substances in blood and detect toxins. Here's your astrocyte. Here's the brain separating everything. Here's the blood, and these tight junctions are here, adhering the cells together. Here's the choroid plexus, which we'll cover next. And here's the fenestration. So it's like a little gap, a little window. So the ependymal cells they can range from either simple cuboidal or columnar cells, and they line the ventricles of the brain and the central canal of the spinal cord. The basal surface doesn't actually rest on the basement membrane. It extends processes into the neuropil that merge with the astrocytic processes. And the apical surfaces have the cilia to kind of like move things along in the CSF and microvilli in order to kind of move substances inside of them or like move around. They're joined by desmosomes and gap junctions, and the modified ependymal cells are in the choroid plexus, and they form the choroidal cuboidal epithelial cells, which will secrete the CSF. The choroid plexus is specialized tissue found in the ventricles, and it consists of folds of vascularized pia mater covered by a layer of choroidal cuboidal ependymal cells. The core of the loose connective tissue of the pia mater has a network of large, thin-walled, fenestrated capillaries that will facilitate the exchange of waste and nutrients. And the fenestrated endothelial capillary cells will allow the free movement of some small molecules, and that is particularly dangerous because an infection can spread from like the ependymal lining to the walls of the ventricle and then straight to the brain tissue. The choroid epithelial cells have the microvilli that will project from their apical surface, and those are bound by the tight junction to form the blood-brain barrier or the blood CSF barrier. And these cells have a lot of mitochondria, and they secrete bicarbonate, sodium, chloride into the CSF to allow the osmosis of water. So the water will flow in the direction of these ions, and that will help in the entire circulation of the CSF. And then the CSF is absorbed back into the venous circulation by the arachnoid villi. Oligodendrocytes are myelin-forming cells. They are the predominant type of neuroglia found in the white matter. They form layers and then they wrap around the parts of the axons, except for at these nodes of Ranvier. And one oligodendrocyte can myelinate up to 50 different axons. And several of them make the myelin internodes, also known as the nodes of Ranvier. So the oligodendrocytes, here they are, they have relatively little cytoplasm and abundant Golgi apparatus. They have round nuclei, and the processes aren't seen in the H&E stain. Here is the neuronal cell body, or the soma. So the glioma are glial cell tumors. Astrocytoma are the most common. It's a slow progression due to the compression of the brain structure. So you can see here, look how it pushes in. And it's characterized by the presence of GFAP. And the only way you can really diagnose it is when a patient comes in complaining of like strange neurological symptoms, and you have to MRI it, because depending on where the glioma grows, it can affect a whole lot of different things. So if somebody has a glioma growing here, their frontal cortex will be affected. Back here would be their occipital. Right here seems like the temporal parietal. So this person will have balance problems, along with some hearing problems. The polycytic astrocytoma is the least malignant, so those will not spread, or those will not tend to spread. The glioblastoma are more malignant, and they're less differentiated. They tend to be more in the frontal and temporal lobes, like this. And the oligodendrocytomas are the rarest. They are a rare tumor of the white matter. And usually those are located in the temporal lobe, and that will cause epilepsy. And then ependymomas result in hydrocephalus. So to differentiate between gliomas and hydrocephalus is like looking on an MRI. A glioma is made out of tissue, and it's going to appear white. Hydrocephalus is made out of like too much CSF, and liquids are black on the MRI, and hydrocephalus will really only stay where the ventricles are. Look. Here's a difference of all like the different cells. Here's the astrocyte. All these like long processes. Here's the oligodendrocyte. It's kind of small. You can't really see the processes at all. Here's their cytoplasm. And then here's the microglial right over here, like the smallest ones of them all. Okay, so the meninges, they protect and they support the brain or the spinal cord. The dura mater is also known as the pachymeninge, and it's made out of a dense fibroelastic connective tissue, and it's lined on the inside by flat epithelial cells. The arachnoid mater is the thicker layer of loose fibrocollagenous connective tissue covered by simple squamous epithelial cells on like the superior surface and the inferior surface. And those lack the capillaries, but they do have large vessels, as you can see here. It kind of runs right through the vessel, or the network. And those are interconnected to the pia mater by a network of collagenous fibers. And then the arachnoid villi will penetrate into the dura. The pia mater is a delicate layer of squamous epithelial cells. Those are not in contact with nervous tissue. Instead, they're separated from the nervous tissue by a layer of astrocytic end processes that form the glia limitans. The glia limitans is also playing a role in the blood-brain barrier. So the pia mater and the arachnoid mater is actually continuous. Some older books kind of call them as the leptomeninges. And the blood vessels that penetrate the CNS go and penetrate them via the perivascular spaces. And the capillaries are not covered by the pia mater, but they are like covered and lined by epithelial or endothelial cells. Okay, that's it for now. Thank you for your attention, and good luck studying.